The overall goal of the following experiment is to express target proteins transiently in tobacco to gain an idea about the feasibility of plant-based enzyme expression in a short time frame. This transient expression is achieved by infiltrating tobacco leaves with a strain of Agrobacterium tumefaciens, which contains a vector incorporating the gene of interest. As a second step, after incubation, infiltrated plant leaves are harvested and protein extracts are partially purified to obtain a working enzyme solution. Next, protein analysis is performed in order to assess the effects of plant-based expression on the enzyme and to verify its enzymatic activity. Results are obtained that show protein size, possible glycosylation effects, and enzymatic activity based on Western blotting, zymography, and substrate degradation assays.
This method can help answer key questions about plant biomass conversion and can also provide insight into the activity of enzymes after being transiently expressed in tobacco.
Visual demonstration of this method is critical, as the infiltration steps are difficult to learn due to the leaves being easily damaged by the syringe. Holger, Klose and I will be performing the experiments.
Under sterile conditions, transfer single colonies of Agrobacterium tumefaciens to an Erlenmeyer flask containing 10 milliliters yeast extract broth with antibiotics. Then, incubate the inoculated broth for 36 to 40 hours at 26 degrees Celsius with 100